Hey there, fellow knowledge seekers. Ready to dive deep? Today, it's all about drug elimination.
It's pretty fascinating stuff.
Basically, how our bodies, get rid of the medications that we take, kind of as a behind-the-scenes tour
Yeah.
of the body's waste management system,
Yeah.
but specifically for drugs.
Yeah.
We've got some great excerpts from a biopharmaceutics and pharmacokinetics textbook, ready to dissect, and I'm really excited to have an expert here
Thanks for having me.
to help us break it all down.
Yeah, absolutely. It's a complex topic.
It is. So where do we even begin with
Hmm.
this whole elimination adventure? I mean, we hear a lot about the kidneys,
Mm.
filtering waste and all that, but is that the whole story?
It's definitely a big part of it,
Yeah.
but there's more to it than just the kidneys.
Well-
Before we jump into that, maybe let's take a step back
Mhm.
and think about what happens when you take a medication.
Okay, so I swallow a pill, and then what?
Right. So that pill, or whatever form the drug comes in, that's called the drug product. And it has to perform in a specific way to release the drug, either for a local effect, like, a topical cream, or a systemic effect, meaning it acts throughout the body.
So the drug products is like the delivery vehicle,
Exactly. Yeah.
and then biopharmaceutics would be the study
Yeah.
of how that vehicle gets to its destination.
You got it.
Okay.
It's really about understanding how a drug's properties, the way it's formulated and the way it's administered, all come together to influence how much drug actually gets absorbed into your system.
That makes a lot of sense. It's not just about the drug itself, it's how it's packaged and delivered. I bet the FDA plays a big role in all this.
Oh, yeah, for sure. They have strict guidelines for drug approval, and biopharmaceutics is absolutely crucial for making sure drug product quality and safety are up to par.
Okay.
But once that drug is absorbed into the system,
Mhm.
then we move into the realm of pharmacokinetics, which is what the body does to the drug.
Okay, so pharmacokinetics is tracking the drug's journey through the body.
Yeah, that's a good way to put it. And it's really important for understanding how much drug reaches its target, how long it stays there, and ultimately how it's eliminated.
Makes sense.
And that's where we start to see the interplay between what the body does to the drug, that's pharmacokinetics,
Mhm.
and what the drug does to the body, that's pharmacodynamics.
Okay, so pharmacodynamics is like the effects, good or bad.
Precisely, yeah. It's about understanding how the drug interacts with its target in the body to produce its therapeutic effect, but also any side effects or toxicity.
So to get the best results and minimize the risks,
Mm.
you really need to understand both sides of the coin, what the body's doing to the drug and what the drug is doing to the body. It's like a delicate dance.
Yeah, that's a great analogy. It's like a carefully choreographed dance.
Okay.
Clinicians use this to figure out the right dose, the route of administration, and how often to dose each patient.
This already feels very personalized.
Yes, it is very much so.
Like everyone's body might handle a drug a little bit differently.
You're right. And that's why understanding this individual variability is so crucial, especially for potent drugs that have what's called a narrow therapeutic window, meaning the difference between an effective dose and a toxic dose is small.
I see. Okay. In those cases, a tiny variation in how the drug's handled could be a big deal. So are there ways to actually monitor how someone's responding to like a particular drug?
Yes, absolutely. There's a field called clinical pharmacokinetics, which uses these principles to look at individual patients.
Okay.
And therapeutic drug monitoring. That's where clinicians can adjust doses based on actual measured drug concentrations in the blood, just to make sure they're getting the best treatment and minimizing the risks.
Wow. So we can actually measure how much of the drug is in their system and then adjust accordingly. That's amazing.
It's an amazing tool. It really highlights the importance of understanding pharmacokinetics to make treatments safer and more effective. But let's go back to that concept of drug exposure for a second.
Yeah, you mentioned that earlier. It has to do with how much of the drug actually gets into the body.
Right. Drug exposure is basically a measure of the dose you take and the resulting drug concentration in your body over time.
Okay.
And this drug exposure is what's directly linked to the drug response or the pharmacological effects you experience.
So more drug means stronger effect.
Not always that simple, but generally, yes.
Okay.
And this is where that concept of the therapeutic window comes in. It's that sweet spot where the drug concentration is enough to be effective, but not so high that it's toxic.
So it's like finding the Goldilocks zone for drug concentration. Not too much, not too little, just right.
Exactly.
Okay.
And that's why understanding drug exposure is so vital for
Mm.
optimizing therapy and minimizing those adverse effects.
This is like a detective story.
Right.
We're piecing together all these clues about the drug's journey and its effects and the optimal concentration.
Yeah.
But what happens when the body decides it's time to get rid of the drug? How does that whole elimination process actually work?
That's where the real mystery unfolds, and that's what we'll dive into next.
Okay, so the drug's done its job, partied hard,
Right.
and now the body's like, all right, time to go. How does that actually happen?
So there are two main ways the body gets rid of a drug.
Okay.
Excretion. That's where the drug is removed intact, basically like escorted out.
Okay.
And then biotransformation, where the drug is changed into a different chemical before being eliminated, like it gets a makeover, before leaving the party.
So two different exits, depending on if it leaves as is or if it needs like a disguise.
Exactly. And for excretion, the kidneys, those are the main players.
Okay.
But there are other routes too, like bile, sweat, saliva, even breast milk.
Wait, breast milk.
Yeah.
So like if a nursing mother is taking medication, some of that could end up in the breast milk.
That's right. That's why it's so important for nursing mothers to talk to their doctor about any medications they're taking to make sure it's safe for both mom and baby.
Oh, for sure. That's a really good point.
Yeah. And don't forget those volatile drugs.
Oh, right.
Like gaseous anesthetics or alcohol. They mainly exit through the lungs. You literally breathe them out.
Makes sense. So many different ways for a drug to exit.
Yeah.
But the kidneys seem to be handling a lot of it.
They do.
So tell me more about their role in all of this.
So think of the kidneys as these amazing filtration systems. They process about 180 liters of fluid every single day, filtering waste products from the blood and producing urine.
180 liters. That's a lot.
That's like a whole swimming pool.
Yeah. But I guess they don't just dump everything out, right?
Right. They're very efficient. They reabsorb about 99% of that fluid to keep the body's fluids in balance. But that stuff that needs to go, like many drugs, gets sent out with the urine.
So drugs are caught in that 1% that gets eliminated. But how do they decide what to keep and what to toss out?
That's where it gets even more interesting. There are actually three main processes involved in renal drug excretion.
Okay.
Glomerular filtration, active tubular secretion, and tubular reabsorption.
Okay, three different mechanisms, each with their own set of rules, I'm guessing?
Exactly.
Break it down for me. Let's start with glomerular filtration.
So imagine a sieve. That's basically what the glomerulus is. It's this network of tiny blood vessels in the kidneys,
Okay.
and it allows small molecules, including lots of drugs, to pass through from the blood into the urine.
So it's like size-based.
Yeah, pretty much.
Smaller molecules get through, bigger ones stay behind?
You got it.
Okay.
And here's another key point. Drugs that are bound to proteins in the blood, they're too big to be filtered. So only the free, unbound drug can actually slip through that glomerular sieve.
So it's not just about size. It's about, like, relationship status
Yeah,
with those proteins in the blood.
that's a great way to put it.
Okay, what about active tubular secretion?
Okay, so this is where those transporter proteins come in.
Transporter proteins. Those sound familiar, vaguely,
Yeah.
from biology class. Remind me what they do.
Think of them like tiny bouncers in your kidneys.
Okay.
They grab onto certain drugs and actually pump them from the blood into the urine, even if the drug is trying to like sneak back in.
So it's not just passive filtering. They're selectively choosing which drugs to kick out.
Yeah. And here's where it gets really interesting. These transporters, they can get saturated. It's like having a limited number of bouncers at a club.
Oh.
If too many drugs are competing for the same transporter, it can actually slow down elimination.
Oh, wow. So it's like musical chairs for drugs.
That's a great way to think about it.
Okay.
Now, for the last process, tubular reabsorption. This is where things can get a little tricky, and where that pH factor you mentioned comes in.
Okay, pH. I knew it was going to make an appearance.
Yeah.
So remind me, what's pH all about again?
So pH is a measure of how acidic or basic a solution is. In this case, we're talking about urine.
Right, right. But how does the pH of the urine actually affect drug reabsorption?
Okay, so imagine the drug molecule. It has these little handles. And the pH of your urine, it can change the shape of those handles,
Okay.
making it easier or harder for the kidneys to grab onto the drug and reabsorb it back into the bloodstream.
So acidic urine, basic urine, it's like changing the grip.
Yeah, exactly. And why that matters is if the kidneys can't grab onto the drug, it stays in the urine and gets eliminated. But if it can grab on, the drug gets reabsorbed and stays in the body longer.
So changing the urine pH could actually change how fast a drug is eliminated.
Exactly. That's why things like diet, certain medications, even the time of day, which can all affect urine pH, can impact drug reabsorption.
Wow, that's wild. So many factors at play here. But before we get lost in the pH maze, can we talk about clearance? You mentioned it earlier. You said it's a key parameter in pharmacokinetics.
Yeah, absolutely. Clearance is basically a measure of how effectively the body is removing a drug
Okay.
from a volume of fluid, usually
Yeah.
plasma, over a certain time.
Yeah.
Think of it like how quickly the body's cleaning crew is working to get rid of that drug.
Gotcha. So a higher clearance means a faster cleanup crew,
Exactly.
and the drug leaves the system quicker.
And here's why it's so important. Clearance directly relates to drug exposure. That balance between dose and concentration we talked about.
Right.
We want to be in that therapeutic window. Effective, but not toxic.
For sure. So understanding a drug's clearance helps you figure out the right dose to hit that sweet spot, minimizing the risks while you're maximizing the benefits.
You got it. It's like a roadmap to guide those dosing decisions.
I like that. And you said that clearance is even more useful than half-life in this context. Why is that?
Because clearance considers all the elimination processes, metabolism, excretion, everything. Half-life only tells you how long it takes for the concentration to decrease by half. It doesn't give you the whole picture.
Makes sense. Clearance is like a more holistic measure
Exactly.
of how the body's getting rid of the drug. But how do you even measure clearance?
So there are different ways to calculate it depending on the situation. One common method involves looking at the dose of the drug and the overall drug exposure, which is measured as the area under the curve, or AUC.
Ugh. AUC, we've seen that before. It's like the total amount of drug that enters the bloodstream over time. Right?
You got it. And the relationship's pretty straightforward. Clearance equals the dose divided by the AUC.
Okay.
So this formula can actually help determine the dose needed to achieve a specific target exposure.
So it's like a math equation that helps personalize treatment.
Exactly. That's the beauty of understanding pharmacokinetics.
Yeah.
And remember, we can also zero in on the kidney's contribution to all of this by calculating renal clearance,
Okay.
which tells us how much drug is specifically cleared by the kidneys.
So we're focusing on the kidney's efficiency in removing the drug. But I'm guessing there's more to this renal clearance puzzle,
There's
right?
always more.
So how exactly is it calculated?
Let's unpack that. To calculate renal clearance, we need to look at a few key factors.
Okay, hit me with those factors. Yeah, hit me with those factors.
So it's like we're doing a forensic investigation. We need to analyze the evidence. And in this case, that evidence comes from the urine and the plasma. We need to know the drug's concentration in both to get a clear picture of what's happening.
So we're comparing the amount of drug that's in the blood to the amount that ends up in the urine.
Right. And the ratio of these concentrations, the urine to plasma ratio, that tells us a lot about which elimination processes are dominant. Like if a drug is mainly eliminated through glomerular filtration, and there's no reabsorption or secretion, that ratio should be close to 1.
So about the same amount of drug in the urine as in the plasma.
Yeah. But here's where it gets interesting. If that ratio is greater than 1, that's a clue that active tubular secretion is happening. Those transporter pumps are moving that drug into the urine. So more is ending up there than what was just filtered through the glomerulus.
So a higher ratio means the bouncers are doing a good job.
Exactly. Now, on the other hand, if that ratio is less than 1, it suggests that some of that drug is actually being reabsorbed back into the bloodstream. Remember how we were talking about pH?
Right, the little handles.
Exactly.
Yeah.
So the urine to plasma ratio gives us a quick look into which process is the main driver of renal clearance for that particular drug, whether it's filtration, secretion, or reabsorption.
Gotcha. But we need one more piece of the puzzle to calculate renal clearance, right?
Yeah.
You mentioned urine flow rate.
Yes, urine flow rate. We got to know how much urine is being produced per unit of time.
So it's not just about the concentrations, it's also about how fast the kidneys are flushing things out.
Exactly. And the formula for renal clearance actually combines those factors. Urine concentration, multiplied by urine flow rate, and then divided by plasma concentration.
Okay, so we're basically figuring out how much drug is being removed per unit of time, taking into account the concentration in the urine and how quickly the urine is flowing.
You got it. Now, remember those benchmark drugs we talked about earlier, like inulin and creatinine?
Yeah, the ones used to measure GFR.
Yes. Those are the gold standard for assessing how well the kidneys are filtering. And the renal clearance values. They're often used as reference points.
Okay.
For example, if a new drug's renal clearance is similar to inulin's, it suggests that glomerular filtration is the main way it's being eliminated.
So we're using those known drugs to kind of map out the elimination pathway for other drugs.
It's a clever trick. And if a drug's clearance is much higher than inulin's, that means active tubular secretion is a big contributor. Those transporters are working overtime.
This is like a detective story for drugs.
Yeah.
We're using these clues and comparisons to understand how each drug is being eliminated. This is super cool.
Yeah.
But is it always just the kidneys? What about other organs involved in this elimination process?
That's a great question. So while the kidneys and liver are definitely the heavy hitters in drug elimination, other organs can get involved, like the lungs, the intestines, even the skin.
So it's a team effort.
Right, it's a team effort. And each organ has its own clearance rate, which depends on things like blood flow and the drug's affinity for that organ.
So certain drugs might prefer to exit through the lungs, while others head for the intestines. It's like they have their preferred departure lounge.
I love that analogy. And to calculate this organ drug clearance, we use a similar idea as renal clearance.
Okay.
But instead of urine, we're looking at the difference in drug concentration between the blood entering and leaving that organ.
So measuring how much drug is removed as blood flows through.
Exactly. And we also got to consider the blood flow rate to that organ.
Right. Because a faster flow means less time to hang out in the organ.
Precisely. So the formula for organ drug clearance involves multiplying that blood flow rate by something called the extraction ratio, which tells us how much drug is removed from the blood as it passes through.
So high extraction ratio means the organ is really good at grabbing that drug out of the blood.
You got it. It's a measure of the organ's efficiency.
I'm starting to see the bigger picture here. We've gone from focusing on renal clearance, which is specifically the kidneys, to organ clearance, which takes into account multiple organs.
Right, and it's this interconnectedness that makes drug elimination so fascinating and complex, but it also makes it a powerful tool for personalized medicine.
Right, because by understanding all these factors, we can really fine-tune treatment for each person.
Yeah, maximizing effectiveness while minimizing those risks.
That's amazing. And it's all thanks to those researchers in biopharmaceutics and pharmacokinetics unraveling all these mysteries.
It's true, they're doing incredible work.
This has been an incredible journey. We've explored the ins and outs of drug elimination, from the kidneys to other organs, and we've seen just how important it is for patient care.
And remember, this is just the beginning. There's always more to learn and discover in this field.
So to all our listeners out there, stay curious, keep asking those questions. Knowledge is power, especially when it comes to your health. Until next time, keep those brains buzzing.